Ever wanted to have a watertight argument against evolution? Try these 3 absolute killers for the scientific dictatorship. The first 2 are a couple of golden oldies, while the third is the clincher. First, appearance of design. Second, irreducible complexity. And third, genetic decay. The design argument was first enunciated by William Paley in 1802. His initial argument goes upon these lines. One is walking through the desert and comes across a watch. Does one assume that it came by chance, or did someone create it? Well, logically, it follows that the complicated design within a watch requires someone more complex, or something or someone more complex than the watch. Therefore, if one is walking through the universe and comes across the biological complexities found within living organisms, which are many million times more complicated than a watch, one would have to assume that it was created by an infinitely more complicated designer. The usual argument offered to counter this logic is that the changes involved in biological evolution are only one step at a time, and therefore do not require the formation of the whole watch at one time. We'll shortly deal with the logical errors in this argument with the concept of irreducible complexity. However, there is one step in evolution that does not have the potential for one step at a time: the first cell. This cell is not subject to natural selection. The closest analogue we have to the hypothetical first cell is a bacterium called Mycobacterium, which has over 580,000 base pairs, i.e. bits of information or letters, and 525 genes. In 2010, a group of scientists tried to computer model a replicating Mycobacterium. In order to successfully model the processes that they knew of, it required the combined computing power of 128 computers running simultaneously. That is no simple watch. It is like walking through the desert and coming across a factory that is automatically assembling fighter jets and believing that it happened by mere chance. However, according to evolutionists, this is not a problem, as time overcomes all problems, even if logic does not. The second argument, irreducible complexity, was first coined by the biochemist Michael Behe. Simply put, novel functions or organs require the simultaneous production of multiple elements that are designed specifically to work together. They are only advantageous to the cell as a working unit, and therefore cannot be formed by natural selection in a stepwise process which requires each step to be advantageous. Michael's example was the bacterial flagellum. This is a tail-like structure on a bacteria that allows it to swim. It is run by a highly complex rotary motor, which is a spectacular example of nanotechnology. Each component or protein of this motor had to be designed specifically for its purpose, and to fit specifically into its adjoining proteins or components of the motor in order for the tail to swim and therefore the whole unit to be useful to the organism. Of course, the usual cry of debunked rings out from the scientific elite. They claim that the motor on the flagellum somehow came about through the combination of other bits of the cell and magically, poof, suddenly worked as a motor. Multitude of steps required to change both desired function and produce the required components are glossed over with words such as ion channel, secretory systems. Once again, time and change reign supreme in the mind of modern man, despite logical argument. The third argument, however, is the deal breaker. Its logic is so overwhelming, the evidence so clear and so repeatable, its predictions so accurate, and the counter-argument so difficult to muster, it brings even the most ardent evolutionist almost to breaking point. It is genetic decay, otherwise known as genetic entropy, most eloquently and succinctly described by John Sanford in 2005. It is the description of the unstoppable and increasingly measurable decay of information within the genome through the process of accumulated mutations throughout generations. This process not only affects humans, but it affects other organisms in which it has been measured. Over the last 5 to 10 years, we have been able to measure the number of mutations accumulating within the human genome. The number is between 75 to 175 mutations per person per generation. Why does this matter? Claims are statistically ignorant. The problem is that out of our 3 billion letters, the majority have a function. When I was going through university, we were often told that most mutations do not matter because only 4% of the DNA was functional and the rest was junk. Junk DNA, like the parathyroid gland, the thymus gland, and the appendix before it, were thought to be non-functional because evolutionary theory predicts much waste. It is becoming increasingly clear that this junk DNA happens to be the most important part of the DNA, as it controls the expression of the rest of the DNA. The ENCODE study in 2006 claims that as much as 96% of the DNA is functional. Thus, 96% of mutations are likely to be affecting functional DNA. Why does this matter, claims those who believe mutations are like mini-gods? Don't they produce the variability required for natural selection? To answer this question, one must think statistically. Think of the DNA as an encyclopedia. If you were to randomly change one letter in any sentence of that encyclopedia, would it be likely to add useful information to you in regard to the topic at hand? Clearly the answer is no, extremely unlikely. However, the DNA is even more complicated than an encyclopedia because it can be coded up to 5 different ways. Thus, one letter in the DNA can be involved in up to 5 different functions. Imagine changing that one letter and it being advantageous for all those 5 different functions. Clearly the answer is almost impossible. Even evolutionists admit that only 1 in a million mutations are likely to be beneficial. The problem is the small detrimental change affected by one mutation is unlikely to be removed from the DNA by natural selection because the effects of upbringing, place of birth, disease and random accident dramatically overpower the 1 in 3 billion change in your DNA. And each generation is producing another 100 of these mutations per generation. They are almost all detrimental and almost all are unable to be removed from our DNA. It is like rust on a car. It is inevitable. Time to give some more concrete examples. Let's start with mitochondrial DNA. Most of the human DNA is found in the nucleus of the cell. However, the mitochondrial DNA is found, as its name implies, in the mitochondria, and it is only inherited from the mother. It is made up of 16,569 base pairs, or letters, or bits of information. Comparing the mitochondrial DNA from humans all over the earth, it is possible to identify the original mitochondrial DNA, often called mitochondrial Eve. The errors, or mutations, are therefore reasonably easy to identify. The average number of mutations in mitochondrial DNA is 22 per person, but some individuals can have up to 100 mutations. Comparing 100 mutations or errors within this 16,000 letter mitochondrial DNA, that means there's an error of 0.6% over human lifetime. Given that we know that the maximum number of mutations within the mitochondrial DNA is 100, all we now need to know in order to determine the number of generations from Eve, or mitochondrial Eve, is the error rate per generation. There are at least 2 studies that have directly measured the error rate within living people, and that number is 1 error every 2nd generation, i.e. your grandchildren will have on average 1 more mitochondrial DNA error than you. This contradicts mainstream analysis, as they only use error rates based on pre-assumed ages, i.e. the length of time required for evolution from ape to human, for example. They do not use directly measured error rates. Therefore, if the maximum number of mutations is 100, and they occur every 2nd generation, and assuming a child is born every 30 years, that would mean there are 6,000 years between us and Eve. If we, however, take the average number of mutations of 22, that would give us 1,320 years from Eve. Error rates must vary, but they will not vary enough to get us 200,000 years, which is what is trumpeted by the evolutionist. Even more amazing, mitochondrial DNA has 3 main lineages called M, N and R. M and N are separated by 8 errors, and N and R are separated by 1 error. How can there be 3 main lineages in the mitochondrial DNA based on the type of errors that are in them? Well, that would mean that following the mitochondrial Eve DNA, i.e. the first original mitochondrial DNA, there was a population explosion leading to mitochondrial DNA error rate. And then there had to be a population bottleneck leaving 3 females alive and therefore 3 quite separate mitochondrial DNA lineages. Well, this happened. How many females of reproductive age were on Noah's Ark? There would be only the 3 daughters-in-law of Noah. This is absolutely amazing confirmation of biblical history, which is etched forever in our DNA. On to the next concrete example, the Y-chromosome, which is only passed down from man to man. There are approximately 59 million base pairs or letters within the Y-chromosome DNA. Comparing Y chromosomes from around the Earth, we find there are 300 mutations in the Y chromosome DNA. The average error rate has been measured at 1 error per generation. That would give us 300 generations since Adam, and assuming a child is born every 30 years, that would give us 9,000 years since Adam. Interestingly, there isn't a large difference between these error rates, as all Noah's 3 sons have the same Y chromosome as their dad. When these same criteria are applied to the DNA of the roundworm, the fruit fly and the water flea, the accompanying error rates always lead us to working out that these organisms are around 10,000 years old. Another fascinating bit of information is that while the Y chromosomes have mutations that are conserved, i.e. almost the same within the same geographical area, the mitochondrial DNA seems to be distributed randomly. That would mean that sometime in the past the human population was split based on generations of related men and not on generations of related women. Did this ever happen in biblical history? Well, it did. We are told that the population was split after the Tower of Babel event into groups based on related men or paternal ancestry. Another biblical event finally etched into our DNA. Genetic entropy and decay not only serves as a fascinating confirmation of biblical history, it also predicts increasing disease, increasing cancer, and reduced intelligence. The scariest thing it predicts, however, is eventual error catastrophe, a coming time in human future where there will be too many errors in the DNA for humans to reproduce, leading to eventual extinction. This is a mathematical certainty without genetic cure or divine intervention. At this stage, the poor evolutionist will be wondering what hit him. Of course, he will never admit defeat, well, some eventually do, but at least he will have something to think about over the coming week.